Logarithms are also used in biology to cope with dynamic range. Some examples of the use of logarithms in biology is things like bacterial growth. As discussed before, when we have experiments with a large dynamic range, we can use logarithms to actually communicate that data in a more succinct way. For example, with bacterial growth, we have one cell which divides into 2 cells, 2 cells into 4 cells, and 4 cells into 8, and so forth. We call that sort of growth exponential growth or logarithmic growth. And when we actually plot that data over time, we will use our log graph paper to present that data to other scientists. The other area where logarithms are used a lot in biology is, of course, the pH scale. And again, and I'll be going through some examples of this in the questions, you will see that when we talk about a pH range from, say, 2 to 14, we are talking about a huge dynamic range in the concentration of hydrogen ions. And again, we use logarithms to be able to condense this sort of dynamic range into a way which can be communicated easily. Another example of the use of logarithms is actually the rate of progression of DNA molecules through things like agarose gel via electrophoresis. In electrophoresis of DNA molecules actually will migrate through the gel at a rate which is inversely proportional to the log of the number of base pairs of the DNA molecule. So when you see those lovely fluorescent barcode-like gels which are shown in shows like CSI and things like that, they are simply DNA fragments which have moved through the gel in at different rates depending upon the log of the number of base pairs of the DNA molecule.